Hello everyone, my name is Thắng Lợi, and I will going with the introduction. And this part will include the history, what define the complement, the component, and the function of the complement. And the first is the history. In late 19th century, the theory Mechikov proposed that phagocytes in the blood could engage and destroy the invading bacteria, thus providing the basis in innate cellular immunity. And in 1891, Buncher and Schlager found heat labile factor in blood that was capable of killing bacteria, and they named it alexin. It means to ward off. And Jules Bordet support this humoral theory by demonstrating that immune disease requires presence of two factors. One is the heat labile lytic factor, similar to alexin, and the second is the heat stable factor, which we know now was antibody. And Paul Ehrlich described that the side chain theory of the antibody formation, especially the mechanism of antibody neutralization by toxin that induce the bacterial lysis with the help of the complement. And according to his theory, the immune cells contain receptors that could recognize antigen, and following the immunization, these receptors multiply and were shed into the circulation as ambulatory receptor. Now we call it is antibodies. And so what is the complement system? Complement system is the part of your body's immune system, and it clean up damaged cells, help your body heal after you have an injury or you are infected by bacterial, fungus, or parasite, and they also destroy the microscopic organism like bacteria that make you sick. And the complement system is made up of the large number of distinct plasma proteins that react with one another, opposite pathogen, and induce the series of inflammatory responses that have to fight infection. And a number of complement proteins are proteases that are themselves which is activated by proteolytic cleavage. And the next one is the component of complement. As you know that there are three distinct pathways through which complement can be activated on pathogen surface. That are alternate pathway, classical pathway, and lactic pathway. These pathways depend on different molecules for their initiation, but they converge to generate the same set of effector molecules. And the component of the complement system include antibodies that are IgM and IgG. They have a C from C1 to C9, and each of protein C1, C2 to C9, they have their own small molecules. They have their own biologically active fragment. Like for example, in C1 they have 3 active fragments that are C1q, C1r2, and C1s2. And the next is MISB1, and MISB2. And they also have mannose binding lectin or ficolins. And the last one is factor B, factor D, and factor P. Each of factor they have their own small active fragments. For example, in factor B they have factor B fragment, active fragment BA, active fragment B. We have totally 5 functions of the complement system. The first is the complement receptor connect complement tag pathogen effector cells. You can see the the CR1 is expressed on both leukocyte and erythrocyte and bind with a high affinity to C3 and C5. And after CR1 receptor on erythrocyte bind immune complex, and when the C3b bind to the receptor of the CR1, they will have a complex immune, and it will go through every pathway of the complement, and then they will take them to the liver where they are picked up by phagocyte and cleared. The antigen cell will be died, and will after the complex protein binding to them come and kill them. Because the binding of complement opsonize microbial cell through CR1 on phagocytes results in receptor-mediated phagocytosis and the secretion of pro-inflammatory molecules as IL-1 and prostaglandin. And CR1 on the B cell mediates antigen uptake of C3b bound antigen and leading to its degradation in the B cell lysosomal system and subsequent presentation to T cells. And the second one is the complement enhanced host defense against infection. Complement protein actively engage in host defense against infection by forming the MIC. Many proteins will bind into the membranes of the target cell, and they will form the MIC by opsonizing potentially pathogenic microbes and by inducing an inflammatory response that has to guide leukocytes to the site of the infection. And after the proteins, most of proteins, they bind into the target cell they bind to the membranes of the target cell. They will forming the MIC and inducing and respond and have a signal to guide the leukocyte to the site of the infection and kill them. And the third is the complement mediators, the interface between the innate and adaptive immunities. After the B cell, they recognize the antigen, they will convert into the plasma cell and create the antibody. And then they will bind to the receptor of the target tissues, and the macrophage will come in and eat them. And then they will and eat them, digest them into small molecules, and take them into the antigen presenting cells. And during the process of antigen recognition and signaling through their respective receptor act to enhance antigen uptake. And they will release the protein cytokine to guide the other immunity cells to come and kill them. And the next one is complement aids in the contraction phase of the immune response. In this, the apoptotic cells express the phospholipid and phosphatidylserine on the exterior surface of the plasma membranes, and then the apoptotic cell will come and bind them through the signal. And the macrophage will come and find them through the signal. And then after binding, they will eat the apoptotic cell. And after binding, the phagocyte will eat the apoptotic cell and digest them into the small molecules. These small molecules will be binding to the cell, binding to the antigen presenting receptor, and then it will induce some immunity cells come and kill them.
Hi everyone, my name is Minh and I represent about the complement pathway part. First, we go to the complement activations is known to occur through three different pathways. First is classical pathway, Latin pathway, and alternative pathway. Three of pathway is involving protein that mostly exists in active zymogen that they are sequentially cleave and activate. All the pathway converge at C3, which is the most abundant complement protein found in the blood, and the result in the formation of activation product is C3A, 3A, 3B, C5A, and the membrane attack complement is called MIC from C5b to C9. And system is a cascade of enzymatic reaction with required uh, spontaneous hydrolysis of the internal C3 thioester and the components of this pathway include the C3, the C5 to the C9. The factor B, D, and P is called the protein. Next, we go to the alternative pathway of the complement system step. Step 1, we have the compound anchoring foreign substrate. The 3D 3B protein binds directly to the substrate and unlike the classical pathway, does not require the formation of antigen-antibody complex. And the step 2 is a hydrolyzed thioester domain of C3 undergo structure change, the exposing the binding site for the factor B. Mg2 plus is the only ion required for functional activation of the uh, alternative pathway that stabilize the uh, interaction between C3b and factor B. In step 3, we have the C3 bound with factor B is cleaved by serine protein factor D from forming C3BB. It's called C3 convertase. CT convertase in the alternative pathway is unique as it is made for the subunit C3b and BB. This complex now can now activate more C3 as a powerful application cycle. Therefore, this pathway is also called the application loop. Next up, we have the convertase complex in turn um, cleave the C3 to C3A and C3B. The 3A fragment is cleaved into the flood phase. Um, and then when second C3B adds to it, the C5 convertase is synthesized. And the last one is the membrane attack complex called MIC is formed following the same pathway as the classical pathway of the complement system. The C3 convertase cleave the, the C5 convertase cleaves the C5 into two units is C5A and C5B, and then the C5B rescue the C6 to the C9 from MAC that induce the cell release. And the application and significance of alternative pathway is this pathway monitor pathogen infection in normal physiological condition. The factor B is a phage protein and increase during inflammation. The 5A and the 3A's are important mediator of inflammation. And the last one is the cell lysis and inflammation are induced by MAC. Next, we go to lectin pathway. Lectin pathway of complement system in the innate immune system include a cascade of enzymatic reaction in which carbon hydrate binding protein called lectin initiate the reaction after binding of the surface of a wide range of pathogens. The pathway is based on the activation of mannose binding lectin associated serine proteins is called MASPS, which are solute serine proteins of serum. And this component of pathway include C5 to C9. The mannose binding lectin is called MBL or picoline. The MASP1 and MASP2, the C2 and the C4. Next, we go to the lectin pathway of the complement system steps. First, we have the MLB is structurally similar to C1Q, um, the classical pathway, and it allows binding on many pathogens. The recognition of MLB sugars on pathogen by MLB initiate of reaction like C1Q in the classical pathway. Uh, the MLB is the six-head monoclonal complex with two proteins, zymogen MASP1 and MASP2, homologous to C1R and C1S. And when the MLB complex by to the pathogen surface, the MLB associate the serum protein MASP1 and 2 get activated, which cleave C4 and C2. Um, the MBL form a thermomolecular complex that activate MASP and result in subsequent cleavage of C4. Next up, we have the C4 is cleaved into C4A and C4B. The C4B attached to the surface of the pathogen includes C2 to bind, which is cleaved by MASP2 to form C2B and C2A. The C2A binds with the C4B, forming a complex C3 convertase. Next, the convertase complex in turn cleaves the C3 to the C3A and the C3B. The 3A fragment is quickly into the first phase, and this complex is called C4B 2A C3B. It's called the C5 convertase, um, and synthesis proceeds on the surface membrane of the pathogen. And the last step is the membrane attack complex MAC is formed following is the same pathway as the classical pathway of the complement system. The C5 convertase cleave C5 into the subunit C5A and C5B, and then the C5B rescue the C6 to the C9, forming the MAC that induce cell lysis. And this is the application and significance of lectin pathway, as you can see.
Hello everyone, my name is T. I will continue the complement activation pathway. Next, we will move to the classical pathway. This pathway includes some components like C1 complex, C2, C3, C4, and C5. This classical pathway is initiated when the immune complexes are formed after the IgG or IgM binding into the pathogens or to other foreign or non-self antigens. The C1 complex is a multimeric complex which consists of C1, CR, and CS molecules, and then it will bind to the Fc portion of the IgG or IgM immune complex. C1s and C1R will activate as the consequence of C1q binding into the exposed Fc portion of IgG or IgM. And then it will cleave the C4 and C2 to form the C3 convertase, which is called C4B2A. The classical pathway was the first complement pathway to be discovered. This pathway begins with a protein complex called C1. In the serum, C1 exists as a macromolecular complex is made up of 3 distinct subunits, which consists 1 molecule of C1q and 2 molecules each of the serine protease is C1R and C1S. They were held together in a Ca2 plus, which is stabilized complex C2 C1QR2S2 that will cleave the protein, but they are inactive during the normal conditions, so we need to activate it. As you can see in the figure, the C1q will interact with 2 molecules, each of C1R and C1S, to create the C1 complex. The C1q itself is composed of 18 polypeptide chains that associate to form 6 collagen-like triple helical arms, the tips of which will bind to the CH2 domain of the antigen-bound antibody molecule. And C1q can bind directly to some bacterial surface, to antibody bound to pathogens, or an acute phase proteins that also bind to the bacterial surface. The binding of the C1q to CH domain of the Fc region of the antigen complex will induce a change in one of the C1R molecules, and then it will convert in an active serine protease enzyme. And then this C1R will cleave and activate its partner. And after that, the CR protease then cleave and activate to C1S molecules. As you can see in the figure, the C1q will bind to the antigen-bound antibody, and then it will induce a conformational change in the C1R. And then the C1R will activate, and next the C1R will activate the second C1R and order to C1S molecules. The C1S has 2 substrates, C4 and C2. There approximately 90% of C4b is hydrolyzed before it can bind to the cell surface. On binding with C4b, C2 will become very susceptible to cleavage by the neighbor CS enzyme. As you can see in the figure, the C1S will cleave C4 and C2, and the C4 is cleaved first, and then after that, C4b will bind to the membrane that close to C1. After the C4 binds, it will continue to bind to C2, and then it will expose it to the action of C1S. Finally, the C2S will cleave C2 and then create the C3 convertase called C4B2A. The membrane-bound C3 convertase enzyme, which called C42A, now will hydrolyze the C3 and generating 2 unequal fragments. The small C C3A and fragment C3B. A single C3 convertase molecule can generate over 200 molecules of C3B, resulting in at this step of the classical pathway. As you can see in the figure, the C3 convertase will hydrolyze many C3 molecules, and some will combine with the C3 convertase to form a new form called C5 convertase. The generation of the C3B is generally important to many of the actions of the complement. The deficiencies of the complement components that will act prior to the C3 cleavage and leave the host extremely vulnerable to both infectious and autoimmune disease, whereas the deficiencies of components later in the pathway are generally lesser consequence. This is because the C3B will act in three important and different ways to protect the host. As you can see in the figure, the C3 components of a C5 convertase will bind to C5, and resulting it will permit the C4B2A to cleave the C5. That is all mechanism of the classical pathway. Next, we will move to the other pathway called the C5 independent pathway. In addition to the above three pathways, we have discussed the protease released by neutrophil and macrophage. Factors such as plasmin can generate complement activation products. For example, a factor called thrombin is a member of coagulation pathway. It can locally generate C5a in vitro and in C5 deficient mice, which are unable to generate a conventional C5 convertase. As you can see in the figure, there are various pathways of the complement activation. There are data present that above indicate that thrombin has C5 convertase activities. Thrombin has a limited function as a C5 convertase in the C5 deficiency mice, but it will become the dominant C5 convertase in the genetic absence of C3, whether the tissue factor or related factor are activated with the complement activation to convert the prothrombin to the thrombin remains to be determined. So there are a new pathway exists for generation of C5a, especially in the absence of C3, with thrombin acting as a potent C5 convertase. It seems that when the delicate balance between the various components of the complement and the coagulation pathway is perturbed, as in the C3 knockout mice, there exists a compensatory adaptive pathway which features increased presence of thrombin, which can be functions as a C5 convertase to generating an authentic C5a. As you can see, there are a new pathway exists for generation of C5a, especially in the absence of C3, which thrombin will act as a potent of C5 convertase. And that is the end of the mechanism of all complement pathway. Next, we will move to the next part.
Next, we go to the regulation of complement activities. All the biological system with the potential to damage the host by their metabolic pathway, cytotoxic cells, or enzyme cascade are subject to rigorous regulatory mechanism, and the complement system is no exception with this general rule, especially in light of the potent positive feedback mechanism and the absence of antigen specific specificity of the alternative pathway. The host must ensure that the destructive potential of complement protein is confined to the appropriate pathogen surface and that collateral damage to the healthy host tissue is minimized. Below, we discussed the different mechanism of which the host cell protect itself against the potential uh, ravage of inadvertent complement activation. First, we go to the complement activities is passive regulate by protein stability and cell surface composition. Protection of the of vertebral host cell against the complement mediated damage is achieved by both general passive and specific active regulator mechanism. The related instability of many complement components is the first means by which the host protects itself against uh, inadvertent complement activation. For example, the C3 convertase of the alternative pathway C3b BB C3b has a half-life of only 5 minutes before its breakdown, unless it is stabilized by reaction with property. In addition to these more passive environmental break on inappropriate uh, complement activation, a series of active regulator protein act to inhibit, destroy, or turn down the activities of complement protein and their active fragment. Next, we go to the C1 inhibitor. C1-INH promote dissociation of C1 component. As you can see in the picture, we have the C1E and INH. It's called a C1 inhibitor. It's the plasma protein that binds in the active site of serine proteins, uh, effectively poisoning them. Uh, C1-INH belong to class of protein called serine protease inhibitor sub and is act by forming a complex with the C1 protease either C1R2S2 causing them by dissociate from C1Q and preventing further activation of C4 and C2. The C1 inhibitor inhibited both C3b and the serine protease MASP2. It is the only plasma protease capable of inhibitor the inhibition of both the classical and lectin pathway. Is present in plasma serve to control the time period during which they can remain active. Next, we have a decay accelerating factor promote decay of C3 convertase. Um, since the reaction catalyzed by the C3 convertase enzyme is the major application step in complement activation, the general and the lifetime of the C3 convertase, the C4B2A and the C3BB are the subject to rigorous control. As you can see in the picture, is with the classical pathway we have a C4B2A and the factor is DAF, CR1, C4BP is prevent and separate the this complex is two subunits is C4B and C2A like and it's the same for the ethanol pathway. Several is separate the C3BB by DAF, CR1 and factor 8 to the two subunits E C3B and BB. Next we have the factor I declare the C3B and C4B. The factor I is a soluble constitutively active. The serine protease can cleave the membrane associate C3B and C4B into an inactive fragment. The factor I require the presence of the cofactor in order to function, and that's true of this cofactor is called the NCP and CRI effect on the surface of the host cell, but not in the surface of the microbial cell. Uh, the the other two factor is factor H and C4PP is derived above by by host cell surface peptidoglycan. And as you can see, the picture is the subunit C3B is the factor is clip them into the three small little subunit A C3C, IC3B, C3DG, with the I is inactive, and it's the same way for the C4B. Next, we go to the protecting inhibit the NAC attack. The protecting is CD59 by any C5B 678 complex that may be deposited on the host cell and prevent them and prevent their insertion into the host cell membrane. Protecting also block the further C9 addition to develop the NAC. In addition, the soluble complement S protein, otherwise known as the vitronectin, by any blood phase C5b67 complex released from microbial cells, prevent their insertion into the host cell membrane. And the last one, we have the carbon peptidase can activate the anaphylatoxin C3A and C5A. The carbon peptidase are general class in the enzyme that remove amino acids from the carboxyl termini of a protein. The specific enzyme that mediate the control of anaphylatoxin concentration at carboxylase N, B, and R. These enzymes remove the arginine reduced from carboxyl termini of C3A and C5A to the form of the to the form is called the arginine is the active, inactive form in the molecule.
Good evening, everybody. Next is my part about biological consequences. So I will talk about the regulation and the efficient of the complement may affect your human body. So I will talk this regulation complement first. So as we talk about the complement system play a critical role in both innate and adaptive immune response. And clearly, but there are some pathogens can go to great length to avoid complement activation. And the infection in complement protein can lead to some serious consequences, like lupus and HUS. But if the complement system may have as may something like it's activate more and more, it may still lead to some serious disease and pathological disease such as asthma, and hyperaccurate organ rejection. So I will talk more about how the efficient protein in human complement system can lead to Alzheimer's disease. So there are two types of complement protein, include C3 and C1q, are thought to bind to amyloid beta protein in the brain in the early stage of Alzheimer's syndrome. And its subsequent complement mediate lysine of neuron and may further accelerate the syndrome. Okay. Next, I will talk about anaphylatoxin. So anaphylatoxin include two protein, C3A and C58, and it act as a potent of chemical attraction for cells such as phagocyte. There are two phagocyte. Here are neutrophil and monocyte. And leads that cell to the inflammation site. So these two proteins, they act as a vasodilator and induce muscle contraction. Sometimes they can cause histamine release from the mast cell and induce oxidate pulse. It means the consumption of O2 from neutrophil. And that can imply in the production of cytokine. This cytokine are called TNF alpha during the liver generation. Next, I will talk about the efficiency of human complement and biological outcome. I will talk about three main the efficiency. So I will talk first about hemolytic uremic syndrome, also called HUS. So what is mean HUS? It means what is HUS syndrome? HUS syndrome is when the blood in our body breaks down and it will increase the urea in our blood, and it result to form a blood clot, and that blood clot will predominate in the kidney and make the kidney swelling. And so what is the mechanism can lead to HUS? So in the recent research is that we can see that the mutation of the factor H genes result in the impair of C3 protein, also called convertate activation, and it associated with atypical hemolytic uremic syndrome within the hemolytic uremia. So how the factor 8 can lead to that? So factor 8 factor A protein bind less efficiency to two proteins, C3b and C3d, on endothelial cell, and as a result, there is increased vascular damage, especially to the endothelial cell, which results in extravascular deposit of fibrils. So it can lead to HUA syndrome. Next, I will talk about the second division of human complement, called paroxysmal nocturnal hemoglobinuria, also called I'm sorry. I'll call paroxysmal nocturnal hemoglobinuria. So in our complement regulator, we have two proteins called CD59 and DAF, are both membrane-bound glycophosphatidylninositol, also called GBI, are linked molecules that involved in inhibit the MAC complement and causing disorderate of C3 and C5 conservates. So how the mechanism can lead to BNH. The BNH mutation in the gene of BIGA result in decrease of expression of GBI-linked protein, including CT55 and DAF. As a result, intense complement mediated lysis of red blood cell occurred in B and A patients, with consequences of hemolytic anemia. White blood cell can also subsequent to lysis and tissue factor released from this damaged cell and may contribute to thrombosis. So next, I will talk about the last division of the human complement system. So the last one I will talk about angioedema. So what is angioedema? Angioedema is a swelling in the deep layer of the skin and other tissue, and it may lead to itchy and grass. So as you can see in this picture, we can see all the swelling around the eye and the lid, and it may lead to the itchy. So what causes this disease? So the mechanism that lead to disease is involved in the C1NH. NNH, a C1 inhibitor and a factor of 12A, result in dysregulator of bradykinin protein, and which of this product which increase the vascular permeability ability, leading to acrodemia as seen in hereditary angioedema. So HAE can also underscore the interaction of the protein involved in a complement coagulation and contact of proteolytics. And that is all of the deficiency in human complement. So move to our last part. I will talk about the conclusion of our part. So the conclusion, as we talk about, we can see that complement system play a very important role in innate and adaptive immune response, from spontaneity of the pathogens and chemo attraction to removal of apoptotic and necrotic cell. Thank you for your attention, and we hope that we can receive your question to further explain this topic. Thank you very much.